All right, today's topic is Chapter 11, Unit 4, which is all about the membrane potentials. So we are talking about fundamentals of the nervous system, the nervous tissue itself, the different cell types, and this lecture is going to focus on the neurons themselves that you can see here, cute little guys, and their membrane potential. So we have a lot of different receptors, we have a lot of different channel proteins here that are going to set this membrane up with a resting membrane potential that's going to allow, when it wants to, relay signal from point A all the way through the axon to the terminals point B to talk to the next cell in line, whether that's another neuron or potentially maybe a muscle, hormonal tissue, anything like that. So for our neurons, general idea here is that all our cells have different jobs, and the neuron's job is to be extremely excitable with that membrane potential and have proteins built into that membrane to allow for that signal to relay that change in ion concentrations from one end to the other very quickly, rapidly, to relay a signal. All right, now all your cells in your body have resting membrane potentials, muscle cells, everybody. But these guys have very unique control over that and are able to relay that from a electrical signal down to a chemical signal released from these terminals to talk to the next cell in line. All right, so let's talk about that excitability, that electrical stimulus that these cells have. Basic principles of electricity. All right, opposite charges are attracted to each other. Opposites attract. Everybody knows you have a negative, you have a positive. They want to get together to cancel out. All right, we have a membrane that's going to separate these charges and sometimes let them get together and sometimes not. With those opposite charges attracting with each other, you need energy to keep them apart across that membrane. So we're going to have some channel proteins that use a little ATP to push things across their concentration gradients uphill, right, to create that membrane potential, kind of like backing up water behind a dam. You're holding all that potential energy there, that flow of water through. When you open those gates, whoosh, rushes through, spin those turbines, and turns it into the electricity that we use. Well, for these guys, they're going to be able to open those channels, flip that potential, and use that as a relay signal, like people doing the wave in a stadium, to take that signal from one end to the other, and then they're going to relay that out into the chemical energy release, those chemicals like neurotransmitters, okay, to talk to the next cell and allow it to relay the signal as well. All right, so when the energy is liberated, okay, that's when those charges move towards one another. We're going to open up those doors and windows. And when the opposite charges are separated, the system has a potential energy. And that's our topic today, is how do these cells set up that resting membrane potential, that potential energy they can use when they need to use it to talk to the cell downstream from them. So a few definitions on electricity. All right, I'm sure you've heard these terms before. Voltage. All right, voltage is a measure of the potential energy generated by separating the charge, okay, by separated charges. And in your house, you have, your 110 volts, you have your 220 volts if we're running the dryer. For our cells, they aren't that powerful, right? We're keeping some ions separate, but we're down into the millivolts here, not the actual volts. And we create what's called the potential difference, or that resting potential, and that's the difference in charges across the membrane. You're actually going to see that our cells, when they're resting, the neurons, have a more negative charge inside than outside, which means they have more positive ions outside than inside. Okay, the greater the difference, the higher the voltage. And again, our cells, aren't going to shock you to death. They just have a slight millivolt change, and it's actually a millivolt change of -70 when we measure it. So you can use what's called a voltmeter. And if we took, say, a little section here of this axon on a neuron and blow it up, and we're able to stick a little meter to measure the inside voltage compared to with this meter, the outside voltage, you would see the negative charge on the inside compared to the outside is -70 millivolts. That is our resting membrane potential for our neurons. And I'm going to show you by the end how that's all set up. with potassium and sodium ions and a little bit of negative proteins inside and a little bit of negative chloride ions outside. That's where we're going. -70 and how we do it. More into the definitions. We did voltage. Now let's talk about current and resistance. Current, just like the current of a river, right? The flow of that water. So current is going to be flow of the electrical charges, those ions, between 2 points, aka inside and outside of your membrane. And this can be used to do work. If you got a difference, you've got something that can be set up to flip, and we can use that. So that flow is going to be dependent on voltage. Okay, it's also going to be dependent on resistance. And resistance is just that, a hindrance or resistance to that charge being allowed to flow. Now, we can have things that are excellent conductors, like the copper wires in your house, okay, to flow electrical, and also those lovely little neurons you have. We can also have things that don't flow very well or have higher resistance, would be insulation. So the wrappings we put around the copper wires, okay, that's an insulator. It's going to hold that good conductor charge inside. We also do the same thing with our lovely little neurons. Think about those myelin sheaths, right? They don't conduct electrical very well, but they insulate it beautifully by wrapping that conductor we have in a little bit of insulation and allowing that charge to be truer and stronger and run even faster down that line to the axon terminals. So voltage, current, resistance, we can put those into a lovely little calculation, and that would be Ohm's law. Okay, it's going to give a relationship by a calculation here to voltage, current, and resistance. So current, known as I, equals the voltage divided by the resistance. So what that means is current is directly proportional to voltage. We got more voltage, we've got more current. You got more juice, you're going to roll a little faster, right? But it can be inversely proportional by whatever resistance we're giving to that voltage. So if we are not allowing it to flow very well, like damming it up, okay, that is going to decrease our current and slow it down. If we have less resistance, that voltage will directly correlate and we'll have higher current. Okay, so it's going to roll faster if we don't have resistance. Makes sense. Okay, we'll be able to roll from the cell body and the dendrites down that axon to the terminal a heck of a lot faster if we are insulated up. So in our cells, in those cute little neurons, we're going to have some proteins, right? Those channel proteins to allow for charges, ions to roll in and out, because they're not just going to flip-flop happily across that phospholipid bilayer. So we have to make doors and windows for them to get in and out. And those are going to be those membrane ion channels. And we're going to use different kinds, right? There's different types of ion channels, but they're all going to be those integral proteins, those ones that roll through from one side of the phospholipid bilayer to the other. Now, big point on these guys is that open doors and windows aren't just open for everything. All right, they are going to have to be for one specific ion. So you're going to have protein channels that allow sodium. You're going to have protein channels that allow potassium, K plus in and out. Right? They decide what they want to allow in and out. And we have to have different types for different ions. Now they can also be categorized not just by what they allow in and out, these ion channels, but how they allow components in and out. Are these proteins completely always open? Those are going to be called leakage channels or non-gated channels, and they're just continually open windows and open doors. You can also have gated channels. So these could allow sodium or potassium in and out depending on if they're told to be open or told to be closed. And these gated channels, they can be divided into 3 categories that let you know how they are gated. And we're going to go through each of these: chemically gated, voltage gated, and mechanically gated. The first is chemically gated. And these are going to open and close depending on if we have a specific lock and key fit with a chemical that will bind to that channel protein and tell it to be open or close. So think of the relay of signal to the neurotransmitters, okay, to the muscle. That is a chemically gated channel. That neurotransmitter bound, just like you can see here with this chemically gated sodium-potassium channel. We have that little lock and key fit here for the receptor. And if that key, the neurotransmitter, binds, it then opens the channel protein. This channel protein is specific for allowing sodium to roll one direction and potassium to roll another. Chemically gated. Lock and key fit with a chemical. Another option for the gated is going to be the voltage gated. And these aren't going to be held open or closed by chemicals. They are going to be held open or closed by the membrane potential, by what the charge is in the area that they are sitting in that membrane. Right? So as the membrane potential might change and go from negative on the interior to positive on the exterior, if we flip that, this might take these voltage-gated channel proteins from closed to open, and sodium in this case is allowed to roll into the cell when that membrane potential is flipped. Now we're going to use these big time, these voltage-gated ion channels, when we are relaying a signal from one end of the cell to the other. So you're going to see these, both of them. You're going to see them again. The other option for opening and closing a channel is going to be mechanically gated, and it's actually a physical change in shape to that membrane that's going to cause, and that receptor that's going to cause it to open. So think of sensory receptors. You've got pressure and touch receptors in your fingers and your skin, and when those are physically shape changed by mechanics, like pushing, that is what opens and closes these channel proteins. So think of these for sensory receptors, right? Touch, pressure, those are going to be mechanically gated. And we'll use those definitely at the beginning, especially, of a signal. Right? Think of that first image we showed. Here it shows you see a glass of water, but what if this was in your hand and you felt pressure? That signal could then be relayed to the central nervous system, decide what we want to do about it, and relay it out to a muscle. Those are those mechanically gated. All right, so keep in mind that we have the electrical, right, those voltage gated, we have the chemically gated, and those mechanicals. With our resting membrane potential, we're definitely going to tag into these chemically gated and voltage gated. Right. So basic principles are, when the gated channels are open, it's going to allow those ions to diffuse very quickly. And they're going to move along the chemical concentration gradient. So if you have a whole bunch of sodium on one side, not on the other, you know, everybody wants equal elbow room, so sodium's going to run from the highest concentration to lower. But we also have the electrical gradients as well across the membrane. And remember, opposites attract. So if we've got a whole bunch of positive charge on one side, negative on the other, the positives are going to run down that gradient to the more negative side. So we've got two different gradients kicking here with our cell membranes, electrical and chemical. Hence, the electrochemical gradient. The electrical and chemical gradients combined. Right, so we're going to move ions along that electrical current, positive to negative, negative to positive, because opposites attract, and voltage gated, okay? And then those chemical gradients as well. Too much sodium on one side or potassium on the other, they're going to want to equal elbow out. All right, generating that resting membrane potential. Now we know we have ions involved, we have channel proteins involved, different types, leaky ones, gated ones. Here's what we're going to do. Okay, we can measure that, and we know it's minus 70, but who's setting up what? Okay, that resting membrane potential is minus 70. We are more negative on the inside than the outside, so you should already know we got more negative ions on the inside of the cell than the outside when we're chilling. And when it's at that resting membrane potential, another term for that is called polarized. So there are differences in that ionic composition between your ICF, which is shorthand for intracellular fluid in the cell, and the ECF, which is extracellular fluid, so the fluid outside of the cell. Right. Again, measured by that voltmeter. Okay. Resting membrane potential, more negative charges inside than outside. So what are the differences? Who's inside, who's outside? What's causing it to be more negative inside, more positive outside? Sodium and potassium, baby. Right, there's more sodium, which is Na+, in the extracellular fluid, so outside the cell, than inside the cell. And they like hanging out there because there's a whole bunch of chloride ions. Look at that, sodium chloride, table salt. We are saltwater bags, people. Okay, so they hang out on the outside more than the inside. For the intracellular fluid, we actually have a higher concentration of the potassium ions, K+, than in the extracellular fluid. And inside the cell, there are a lot of negatively charged proteins that hold that positive potassium charge inside because opposites attract. Now, it's that potassium that's really going to play the most important role in membrane potential, because we're going to have a lot of channel proteins that are going to allow potassium, okay, to do some movement more than the sodium and allow for that more negative concentration on the interior. So let's watch a video and you'll get a visual on what's going on in setting up that resting membrane potential with all these different channel proteins built into that entire membrane of a neuron. Here we go.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
All right. So with those two pieces of information about the just open leaky channels and then those gated channels, that is how we maintain the resting membrane potential. All right. Difference is those leaky channels. First of all, you've got 25 times more permeable potassium leaky channels than the sodium. You saw those numbers, that ratio, there were a lot more. So more potassium is diffusing out than sodium can diffuse in. So more positive charges outside, guess what? It's going to result in the inside of the cell being more negative. That's what establishes that resting membrane potential. But you saw that it was actually making more negative. It was in the minus 90 millivolts. It is the second layer here, this sodium-potassium pump using ATP, that stabilizes that resting membrane potential at -70 by pumping out 3 sodiums for every 2 potassiums. We're going to be maintaining a -70, as opposed to if we didn't have the sodium potassium pumps, it would be sitting at a -90 for resting membrane potential. All right, that video I just showed is also available in just the PowerPoint, so you don't have to watch this video to watch that one. You can just play that off of your PowerPoint as well. All right, so the next lecture will be on changing this resting membrane potential -70, but I want to just give you a little sneak peek in the next couple slides. Your membrane potential, right, that -70 is going to change if we muck with open windows and open doors, more channel proteins, right? So concentrations of the ions will change when we mess with the permeability of that membrane. And we can do 2 different changes in membrane potential. Okay, we can have, by changing different open channels, graded potentials, or we can have action potentials. Now, graded potentials, we're going to see in an upcoming video, are very short changes in that potential, and they'll come very quickly back to the -70. There's going to be very localized little tickles. So say a cell along this dendrite has another axon coming in. Say we're at this little area right here, and this green neuron is this guy. You might have little tickles from an axon terminal upstream that might just do little graded potential changes, a little tickle, another little tickle. But if we get enough of those tickles to change local spots of that graded potential, we can get to a point called threshold, where we are going to kick off that second type of potential called the action potential. And the action potential is the one that rolls down the axon. So think action, axon, roll down and release neurotransmitter to the next cell in line, whether it's another neuron or whether it is another type of tissue. So graded potentials can lead to an action potential, which is an all or none, straight down the line change in the membrane potential. So we're going to discuss these 2 different types in an upcoming lecture, and how multiple graded potentials can kick off an action potential. Right? We need these, both of them, to be able to receive signals, integrate signals, and send that information to the next cell in line. One last slide here. This is a kicker. So when we do these graded potentials, these little tickles, they don't always have to be a depolarization, or where we move that -70 towards 0 by allowing more of the positive ions inside, right? Those are going to usually lead to producing an impulse that's going to cause the action potential. We can also have tickling of the dendrites in the cell body that are going to cause hyperpolarization, which is actually make it even more negative inside, which is going to make it even more difficult for that cell to relay signal. And sometimes, especially in those integration cells in your central nervous system, we want to make it more difficult to relay a signal, right? That is also a way we can integrate information, is by the lack of sending a signal. Now, I know saying depolarization and hyperpolarization may be a little furry, so let's look at it in an image form here. With the depolarization, right, we're allowing more positive charge into the cell, so we're going to get closer to 0 and be less negative. That's a depolarization. If we get enough of a depolarization signal, then we can kick off an action potential. That hyperpolarization I was mentioning is where we actually make the cell more negative, further away from the ability to cause an action potential and shoot a signal down that axon to the next cell in line. All right, so there's a little prelude. We've created a resting membrane potential. Upcoming lectures will talk about how we take those graded potentials and action potentials and turn them into signaling, which is what our nervous system is all about. Talk to you later.